Chromosome walking. Chromosome walking is another technique that can be used for the screening of libraries. If you remember, we have discussed about the advantage of random DNA fragmentation, that if we will digest the genomic DNA randomly, what will happen is that overlap DNA fragments will be produced. And these overlap DNA fragments, they can be used for the detection of neighboring fragments, because these random fragments, they can overlap with each other. Their ends will overlap, and one of the fragment, it can be used to detect the next fragment by a technique which is called as chromosome walking. In addition to that, we can say that chromosome walking, it is used to detect the gene whose expression or whose function it is not well known, but its genetic location is known, and this technique, it is called as positional walking, where we are going to detect those genes whose function is unknown but location is known. So for this purpose, as I told you, that we must have random DNA fragment in order to produce a library, and one of the fragment, it can be used as a starting point, or it can be used as a probe, so that next contiguous DNA fragment will be detected. Why next contiguous or neighboring DNA fragment is detected? Because in random DNA libraries, the ends of the fragments, they will overlap with each other, so that one fragment, it can be used as a probe for the detection of next DNA fragments, so that in this way we can move along the whole length of the chromosome, and this technique, it is called as chromosome walking. For example, if we have a very large DNA fragment that contain number of genes, और इन genes को A, B, C, D, E and so on कहा जा सकता है, तो इसमें हम क्या करेंगे कि इसकी हम डाइजेशन करेंगे with appropriate endonuclease like EQR1. तो जो फर्स्ट फ्रैगमेंट है लाइक जीन ए का, उसको क्लोन करेंगे बैक्टीरियोफेज लैम्डा में, ताकि हमारे पास प्रोब आ जाए, और उस प्रोब को हम यूज़ करेंगे जो नेक्स्ट डीएनए फ्रैगमेंट है उसको डिटेक्ट करने में. जब नेक्स्ट डीएनए फ्रैगमेंट डिटेक्ट होगा तो उसके एंड को हम यूज़ करेंगे जो सेकंड लैम्डा क्लोन है उसको डिटेक्ट करने में. तो इस तरह से जो नेक्स्ट फ्रैगमेंट का एंड है उसको हम यूज़ करेंगे एज अ प्रोब, so that we can move along the length of chromosome, and this technique, it is called as chromosome walking. It may be explained with the help of a diagram later on, but in case of human genome, the starting point may be a fragment which called as restriction fragment length polymorphism, that may be closely related to some diseased locus. So there are some genes that may be related to certain diseases. They can be used as a starting point for the detection of next clone. Now it can be explained with the help of diagram. Here we have eukaryotic DNA that may be around 80 kilobase in length, and it contains different type of genes like A capital, A small, B, B, C, C small, D, D, E and E. And these genes can be digested into different fragments with the help of EQR1 enzyme, and these fragments, they can then be ligated within the arms of bacteriophage lambda. And then the first fragment, like that corresponding to gene A, it can be used to detect the next fragment. Like it can be used to detect the fragment of A small, and then A small fragment, it is subcloned to detect the next fragment or the gene which is called as the B. And in this way, the B, it is used to clone the C, and C probe, it is used to clone D. So in this way, because of the random DNA fragments that will be produced, they will have overlap ends, and the overlap ends can be used as a probe for the detection of next DNA fragments, and this technique it is called as chromosome walking. Next diagram, it is more simplified. So this diagram also indicated chromosome walking, where we can detect overlap DNA fragments. So suppose this is a DNA fragment here, and this is our target gene, and how we can go towards our target gene. So this is the first clone that have overlap ends with the next clone. First clone, it is used to detect the second clone, and then second clone, it has overlap ends with next DNA fragment. So in this way, the next DNA fragment, because of its overlapping ends, it can be used as a probe to detect the next fragment. So in this way, we can move along the whole length of the chromosome, and because of this reason, this technique it is called as chromosome walking. One drawback of chromosome walking may be that here the repetitive DNA must not be present elsewhere in the genome, because if repetitive DNA is present, so the detection may be confusing. And this technique of chromosome walking, it has extensively used for the detection of different genes in Drosophila, because in Drosophila the repetitive DNA segments, they are not prevalent when we compare it with higher organism.